So with that, we'll get on with the program, and I'd like to welcome Dr. Bruce Martin, who's a plant pathology and physiology researcher and professor at Clemson University. And Dr. Martin is well-renowned and well-published and has many awards for his service in turf research. And again, I want to welcome Dr. Martin, and with that, Dr. Martin, we'll turn it over to you.
Okay, thanks, Jimmy. Good afternoon, everybody. I want to thank Bayer for giving me the opportunity to participate in this webinar series. And this is a subject kind of dear to my heart because I did my PhD work on that. I won't tell you when, but it goes a way back. We're actually not going to cover everything about Rhizoctonia today, although I may mention a couple of things on warm season turf, but the subject today is strictly cool season turf. So be sure to ask your questions as you have them. And if any of you have had some of my seminars in the past, you know, kind of like the history, so I'm going way back here, and we're going to talk a little bit about taxonomy because some of it's important in the language that we're using today in turf. And you can see there that the genus Rhizoctonia was actually erected back in 1815, which was prior to the germ theory. And so the germ theory, of course, refers to Robert Koch and Louis Pasteur and the recognition that microorganisms cause disease. A fellow named Kuhn described Rhizoctonia solana, which most of us know as the cause of brown patch, but you may also know that Rhizoctonia solana attacks many, many plants. And you probably recognize Solanum, S-O-L-A-N-U-M, as the genus of potato. And that's what he described that on in 1858, which is right in the middle of the germ theory and the development of plant pathology. So I guess this fungus is as old as dirt, and the thing that's neat to me about it is we keep learning about it. So from the birth of plant pathology until now, we're still learning about this very common cause of the turf disease. Now that term Rhizoctonia is a genus, of course. Solana would be the species. The genus Rhizoctonia refers to the asexual forms that we normally see in the turf environment. And you know that in the asexual stage, Rhizoctonia exists simply as a mycelium. It does not produce any kind of asexual spore like a conidium or a sporangium or anything else. No spores in the asexual stage. Some forms of these fungi will produce a sexual stage. That's called a teleomorph, or the sexual stage. And I'm going to show you kind of how they correspond, but just let it be known that we don't think the sexual form plays much role at all in the turf environment. Now, a little bit more modern history, at least compared to the mid-1800s, this was one of the first bona fide turf diseases described. In fact, it was the first, if you discount some of the old, reports of fairy ring back in, ancient times. But that in 1914, Rhizoctonia solana was identified as the pathogen of creeping bentgrass by C.D. Piper. And since then, essentially, it's been reported on all turf grasses, to be honest. Back then, Bordeaux mixture had already been developed and was being used, and it was used to control it on turf initially. You can still buy Bordeaux mixtures in garden stores, which is kind of interesting, and it will still control brown patch while it burns your turf with all the copper that's in it. But, up until probably around the mid-'70s, this was kind of the state of the art. From 1914 to the mid-'70s, we thought about brown patch and maybe a little bit of cool weather brown patch, and that was about it. Then we started learning new things, of course. I'm going to kind of cut to the chase here and give you all the punchline, of this whole webinar, but then I'm going to go back and then talk about each one of these diseases with the exception of large patch. But this is pretty much what we know at this point with the different diseases with Rhizoctonia causal agents. Of course, brown patch, we now distinguish brown patch as the disease on cool season turf grasses alone. The causal agents are still Rhizoctonia solani. I'll explain what that is. And it has some forms of this do produce a sexual stage, but it is not readily seen at all in turf. In fact, it's not readily seen in most agricultural systems, except a few where the sexual form actually causes the main disease. And Thanatophorus cucumeris is the name of that sexual stage. I'll explain what these AG designations are. They're important because they designate different strains of Rhizoctonia solani. Now, we also, some of you are probably familiar with large patch, which is another form of Rhizoctonia solani. The causal agent is a different genetic type. The LP stands for large patch, and it's pretty much limited to warm season grasses. The telemorph, if it were to be found, would be in Thanatophorus cucumeris. We also have Rhizoctonia leaf and sheath spot, which is sometimes called leaf and sheath blight. We have forms of this disease that occur on cool and warm season grasses. Today, we'll just talk about the diseases on cool season grasses. And two causal agents are reported as the causes of leaf and sheath spot. This is primarily a very hot weather disease. Causal agents are Rhizoctonia zea and Rhizoctonia rhizi. And here we have a different teleomorph, a different sexual stage that some of you have heard, Waitia. You've heard that term used. And you can see that several of these have the same sexual stage, Waitia cercinata. We also don't think these are important in turfgrass environments, although they have been identified in laboratory cultures from turf isolates. Probably where you heard the term Waitia had to do with Waitia patch, which is really a term we would prefer people don't use. We don't like to see a name of a fungus being used in a disease name, so it's distinguished today as brown ring patch. And also on cool and warm season grasses, although most folks are familiar with it. If they are at all, they're familiar with it on Poa annua primarily. The causal agent is Rhizoctonia cercinata, variety cercinata, distinguishing it from these genetically, even though they have the same perfect stage. Then finally, yellow patch, which also attacks cool and warm season turf, but primarily cool season turf. And we used to call this cold weather brown patch, or cool weather brown patch. Causal agent is Rhizoctonia cerealis, so a different species altogether with a different sexual stage that's only been identified in the laboratory. All right, so that's kind of what we have. Since a lot of this is developed and been recognized and research has been done on it since the mid-'70s until today, I expect in the next couple of decades this list will probably get longer. I'm not going to spend much time on this, but just so that this is why we don't like to use fungus names for diseases. The taxonomists have been busy with this whole group and have even proposed that we change Rhizoctonia solani to Maneliopsis solani and Rhizoctonia zea to Crusorrhiza zea and Rhizoctonia cerealis to Ceratolrhiza cerealis. And this is based on rules of taxonomy that we don't care about as turf pathologists that much, but it's based on morphological differences and genetic differences and a lot of history. Probably you're not going to see that referred to much in the literature, but if you do, that's what it's referring to. Now I'm going to show you some pictures of these sexual stages when they occur. They're basidiomycete fungi, and you'll recognize these basidia and basidiospores here as similar structures that would occur on the gills of mushrooms, which are higher fungi in the basidiomycete class. These are really kind of referred to as racupinate fungi. They do not produce a fruiting body like a mushroom, so the sexual stage when it occurs just forms right upon the mycelium here. So you can see there's some morphological difference with Thanatophorus, with these long pointed strignata and kind of a large basidium, compared to Ceratobasidium, has very round basidium, compared to Wadia, which has a contorted mycelium, which we also see under the microscope with Rhizoctonia zea and some of these others, and real short little horn-like strignata. So taxonomically, that's how they fall out. Now, the reason I'm showing you this is so that you do realize these are completely different fungi. The only thing really they have in common is some common morphology or similar morphology, but they're quite different. And here's an actual photograph that I took under the microscope of the sexual stage of Rhizoctonia solani being formed here. So we would refer to it as Thanatophorus cucumeris. There's the strignata, and then basidiospores would be blown out like balloons on the ends of that. Okay, so that's about all I'm going to say as far as the sexual stage. Since in turf we deal with the asexual forms, the mycelial forms, well, our taxonomy and our recognition is based on those mycelial forms. So we don't have a spore, with certain colors and numbers of cells and things like that to go on. We simply have a mycelium. And if we get Rhizoctonia in culture, it's always brown in culture. It always turns brown. All of the Rhizoctonias will have a very distinctive pattern of hyphal branching and septation, which we can teach just about anybody to recognize. It has a certain kind of a cell wall called a dolichopore septum, which is a little microscopic tool that we use to recognize mushroom-type fungi. Basidiomycetes have this. And Rhizoctonia solani is always multinucleate, and that's important from a diagnostic standpoint also. Part of the reason I'm telling you this is because not as superintendents, you're probably not going to be doing much of this, but you send samples to clinics. And these are some of the things that the clinicians are going to be looking for when they diagnose your diseases. Usually Rhizoctonia solani produces sclerotia, which are hardened masses of cells and how the fungus survives. In fact, that type specimen that occurs on potato, when you're in the grocery store and you look at some baked potatoes and you see these little black dots on the potato skins, those are actually sclerotia of Rhizoctonia solani. And of course they occur in turf habitats down in the thatch. Most of the Rhizoctonia solani strains that attack different plants are actually pathogenic, but not all of them, which is kind of interesting from an ecological standpoint. They never produce a clamp connection. They never produce conidia. They don't produce differentiated sclerotia, which would be like an orange with a rind in the center. They don't produce ropey strands, which are called rhizomorphs. They're always brown in culture. If they're not brown, they're one of those other species like Rhizoctonia zea. And if the sexual stage is formed, it's always Thanatophorus cucumeris. It always has that morphology that I showed you of the sexual stage. Those are the characteristics of Rhizoctonia solani, which is really not much to go on when you think about it. It's a few characteristics here and a lot of negative characteristics. Now these are kind of the microscopic shots of Rhizoctonia. It doesn't really tell you which species are strained. It just gives you the overall shape of the mycelium as it would be growing out of host plant tissue. In fact, over here on this side is where the isolation, the host plant tissue, was plated on agar. And then you get this typical growth of a runner hypha, a main stem basically, and side branches. And then these side branches, when they're young, this would be the younger mycelium, this would be the older mycelium down here. When they're young, they'll tend to come off at 45-degree angles. Then as they mature, the mycelium matures and gets a little older, you see a lot of 90-degree angles. And you see it here in this close-up. And this close-up also shows you these little light dots here on the edges of the cell walls, which is the doliprane. It's basically a tube, a hole in the septum, and you're looking at it from a sideways shot through the microscope there. And here's an even closer shot of Rhizoctonia. Again, you can't tell the species. They all have these characteristics in common, with a main runner hypha here, and then a side branch that's coming off at a 90-degree angle. And with many of them, not all of them, but many of them, you'll see a constriction right here at the point of the side branch. And all of them pretty much produce a cell wall right here near the point of the side branch. So you see this pattern, the 90-degree angle, the constriction, and then the cell wall that's laid down very near the point of the side branch. This one's actually out of focus, but you can barely see one right over here. And that distinguishes these mycelial fungi from Sclerotinia homocarpa because of dollar spot, which is also mycelial, no conidia. But you'll see the dollar spot fungi produce very long, narrow hyphae as well as thick hyphae. It'll have lots of vacuoles and things like that inside the mycelium that you'll recognize. And then you don't see this pattern. You might see a septum put down over here or over here with Sclerotinia. And of course, with Pythium, the other common organism that folks like to learn to diagnose, you don't see any of these cell walls in here. It's xenocytic. So that gives you a little bit of an idea diagnostically what we're looking at when we're looking through the microscope. And we can quickly say it's a Rhizoctonia. Some more shots, some of them you've already seen here, just showing that general pattern. After a while, you look at enough of this, and it just pops right out at you. You don't need the spores to know that you're dealing with a Rhizoctonia. I told you Rhizoctonia solana is multinucleate, and these are just some of the little lab techniques that we might use with light microscopes to stain the nuclei. The typical light microscopes are cheaper microscopes, and you don't have to do anything too fancy to stain these nuclei and see them. But if you really want to see them clearly, we'll use a DNA stain like this DAPI and use a fluorescence microscope, and that's what you're seeing here. Here's a cell wall, here's a cell wall, here's a cell wall. So this Rhizoctonia has 1, 2, 3, 4, 5, 6, 7, 8, 9. It looks like nuclei inside, so it's multinucleate. Rhizoctonia zea, Rhizoctonia cercinata, Rhizoctonia oryzae are also multinucleate, but they're not going to be brown in culture. They have some other characteristics that we can look at and distinguish them microscopically. The other thing I told you that I would explain or attempt to explain is this AG designation, AG1, AG2, AG2-2 LP for large patch. What that refers to is anastomosis group. AG stands for anastomosis group, and this is just a little simple mycological test where a known strain on this side of a petri dish or a slide might be plated out and allowed to grow, and then a second strain that might be an unknown strain is also put on the other side, and they're allowed to grow together. If they fuse, like you see here, and you can even see some attraction of this mycelium, of this hypha to the other strain. If you see this phenomenon, then it turns out, and this has been confirmed with genetic tests, that these strains are related. In fact, they may even be clonal, identical. And if they are, they belong to the same AG. And that's important because without spores and other characteristics, it's given us a tool to recognize the different diseases over all the different plants where Rhizoctonia solani and the binucleate Rhizoctonias cause disease. With our modern molecular techniques, it's basically confirmed this AG phenomenon. And there's a close-up of a bridge between two strains showing what is termed a killing reaction here. You don't need to know about that, but when you see this fusion here, you know that they belong to the same AG. So that kind of research over the years has explained these patterns that we see. And it turns out that the pathogens that cause brown patch in tall fescue, for instance, tend to be of these strains, this AG1-1A, for instance, which is also the same strain that causes a major disease of rice around the world. So we can relate some of that rice research to turf grasses, which can be important. Turns out that this strain here, AG2-23B, Roman numeral 3B, it's what the Japanese call the Matt Rush isolate, and that's what we typically isolate out of creeping bentgrass. So it's all brown patch. It's all caused by Rhizoctonia solani, but we may have these different strains here attacking different hosts. And of course, we have this one that doesn't even attack cool season grasses, except maybe in the seedling stage, AG2-2LP, that causes large patch, and so on down the line. All of these have been reported from turf grass, and I think in all crops we're up to something like 12 or 15 or so AGs attacking different plants. Then, of course, we've got different species, Rhizoctonia zea with the weighty perfect stage, Rhizoctonia rhizi with the weighty perfect stage, and Rhizoctonia cercinata with the weighty perfect stage, now distinguished by these varieties, zea, rhizi, and cercinata. And finally, Rhizoctonia cerealis that causes cool weather brown patch. Okay, so we have all of those, plus some saprophytes that are out there, weakly virulent or avirulent binucleate Rhizoctonias that are also in the turf environment. I'll just show you some pictures of those. They've been studied just a little bit as biocontrol agents. Now, when we're talking about brown patch on cool season grasses, you now know and knew it before, but you know again that this is caused by Rhizoctonia solani, certain strains of Rhizoctonia solani. Pretty much all the cool season grasses are affected. It's a warm, humid, summertime disease. Typically, the optimum temperatures where brown patch is really active is going to be in the high 80s Fahrenheit, and with nighttime temperatures at the mid-60s up to 70 or so Fahrenheit. That's ideal conditions. On low-cut grass, like a putting green, we typically would get a smoke ring symptom, particularly if it's uncontrolled by fungicides. If you got down close with a hand lens or took samples in and looked at them under a dissecting microscope, you would notice that all those symptoms are caused by leaf lesions, where the mycelium is macerating the leaf tissue, causing water-soaked lesions. And it turns out that Rhizoctonia solani is primarily a pathogen of leaves and crowns, but not of roots in turf. In other plants, they're great root rotters, depending on the strain and the host plant. Then, of course, I've already told you we have these different strains that are out there, and these are the primary strains that we see. There are other ones that we occasionally isolate from cool season grasses. Of course, in hot, humid environments where we are trying to grow cool season grasses, such as the transition zone, we might have a pattern like this where the cool season grass is growing great in the spring of the year, and as we approach the hot summertime, the growth rate of that cool season grass goes down, and then we get some recovery in the fall. And of course, the Rhizoctonia solani, I told you, is favored by those warm conditions and warm nights. So it's favored in the summer when the host is disfavored. So right here in the middle is our epidemic, basically. And as most folks know, the best time to control any epidemic is at the beginning. So this might be the place where your initial fungicide applications would be made to get the best control. Some of that's going to depend on where you are and the ability of the turf to recover or not, and also what you can afford. In lawn care, for instance, we might wait till here, to initiate an expensive strobilurin and fungicide application. But without a doubt, just biologically, we would know we'd get the best control if we started here and we controlled it all through this epidemic to the end. Of course, we don't see this too much on golf courses anymore. This is a shot of brown patch. Sometimes these patches will look like they've just appeared overnight, after a heavy rain and after temperatures might abruptly rise. You have those typical ideal conditions. And here's a classic kind of a textbook photograph of a brown patch in bent grass showing the smoke ring here, which is the leading edge of the infections. You're seeing a couple of things. You're seeing actual mycelium here early in the morning when the dew is still on it. You can see the mycelium itself. And but you're also seeing dark turf just to the inside of that silver lining, which is the mycelium, which is basically the water-soaked lesions. Now, this is a bad brown patch. You can tell because the center's already been colonized by algae here, so the ability to recover and heal this up is going to be pretty minimal. This was taken photograph out of my turf plots here in Florence, South Carolina, probably in July, so that explains that. It's a hot place. Compared to that, to this brown patch where you still have a smoke ring here, and you're seeing this dark turf without the mycelium, perhaps later in the day as the mycelium is dried up. But you still have infections and water soaking along the margin here. Of course, you can tell that this turf is going to recover readily from a fungicide spray, curatively. And on tall fescue, if any of you are managing tall fescue and roughs, here's a brown patch in tall fescue. In June or July under those same environmental conditions that we talked about. And this is a great teaching tool to show you the leaf lesions on tall fescue and, to distinguish these from dollar spot where these lesions are really indeterminate. They just continue to expand until they just rot the whole leaves, as you see here. That's why it's called a blight, not a leaf spot. Of course, you can see the mycelium. You can actually see the initial infections taking place there in a little lesion that would look very similar to a dollar spot lesion. You always see this browning along the margins of these Rhizoctonia infections, and that's actually phenolic compounds that the host plant is producing to try to wall off those infections. It's a sign that the plant is trying to withstand the infections, but of course, under those hot, humid conditions, the host plant physiology is compromised, and it's going to lose that battle. Well, some old research by Houston Couch, I like to use it sometimes to explain those patches just suddenly appearing, when you get hot, humid conditions. But it turns out that a lot is going on even at cooler temperatures. So at relatively cool temperatures, this fungus can grow as a saprophyte. And it can germinate and grow from the sclerotial or colonized plant tissue. And you don't see symptoms, of course. It's just growing along, feeding as a saprophyte. At a little bit higher temperatures, it can begin to colonize the lower leaves in the turf canopy. But because these temperatures are also pretty good still for growing bent grass, for instance, you still may not see the symptoms. And yet this could be, the beginning of your epidemic, and it's something to be aware of from the standpoint of your location and whether your turf can recover or not in summertime heat. And then when you get into these really hot conditions, it comes up quickly. And of course, rainfall and prolonged humidity can promote this along with the temperature. Managing brown patch without fungicides implies everything that you can do basically to improve the host health, which includes balanced pH and balanced fertility. Good air movement, fans and things like that down in the south have helped with brown patch, good sunlight penetration. Managing irrigation on the dry side, of course, correcting drainage problems. Nobody does this anymore. They don't over-fertilize in heat stress. And on golf courses, seems like with Bermuda and with bentgrass, we tend to under-fertilize. And even under-fertilizing can prolong some of the symptoms that some of these Rhizoctonia diseases can induce. So remember I said that when we talk about brown ring patch. I think one thing that's helped in the south and in the transition zone has been improved cultivars for heat stress. We don't tend to see brown patch kill turf and turn it to algae like we did when we had pin cross. But of course, if we're going to manage golf course turf and heat stress environments, we're going to need fungicides. Here's just an old national trial that we had in Florence where I was rating brown patch, and the way NTEP likes these rated is actually the higher the number, the less disease, which is not typically the way we rate with plant pathology. So these higher numbers here have less brown patch than pin cross. And you can recognize A1, G2, A4. Doesn't mean these don't get it. They get it, particularly A4, but they just don't get it as bad as pin cross. Now, fungicides, this is just a little short list of some of them that are registered so I can talk about them a little bit. You have different classes like the DMI fungicides, Balaton, Banner, Eagle, the triticonazole formulations, metconazole. All those DMIs are labeled for brown patch, and they all differ really in their efficacy for brown patch. Some of the weaker ones might be Balaton, triticonazole, and I'm talking about just brown patch here compared to metconazole or Eagle are pretty strong, particularly metconazole being very strong for Rhizoctonia. So we have these differences. The dicarboxamides like vinclozolin and iprodone are very good brown patch materials. And daconil. We used to consider these excellent until the strobilurins came along. So the game changed a little bit, I think, in the late 90s when Heritage came along, followed by Compass, followed by Insignia, and now Disarm. All of these strobilurin fungicides are strong Rhizoctonia materials. Thiophenic methyl over here, such as Cleary's 3336 and other T-methyl formulations, are really good for Rhizoctonia solani, for brown patch. They're not so good for Rhizoctonia zea, Rhizoctonia rhizi, and Rhizoctonia cercinata. Polyoxin D, which is now labeled as firm, I believe, instead of endorsed, is a very strong brown patch material, very good. And of course, flutalonil is very good for brown patch. So that gives you some idea. Brown patch is not really a hard disease to control as long as you get on it early. And as much as anything, the choice of these fungicides has to do with how you develop a disease control program, and there are other characteristics, such as thinning of turf and heat, where you may get away from the DMIs and use more chlorothalonil, or you may use some of the new mixtures with DMIs and chlorothalonil, which mediate some of that. Many fungicides are labeled for brown patch, and the reason I had that simpler slide was to give you some of those high points, but here's an example of 3 slides here, just all these different formulations. Some of these fungicides like Terranebrin we're not likely to use those down in the south because they're phytotoxic. Lots of chlorothalonil formulations and lots of mixtures now that are coming on the market, like Renown or Disarm C, as well as others. I've got 3 slides of Reserve here from Bayer, very good. I like these chlorothalonil triticonazole mixtures. So anyway, you know that there's lots of fungicides labeled for brown patch, and lots of formulations of T-methyl here. Okay, so that's kind of an overview of brown patch. We'll talk about yellow patch now, or cool weather brown patch. This one was known back in the early 70s, early 80s and all. It was called cool weather brown patch. And then Lee Murphy distinguished it with the term yellow patch to distinguish it from brown patch caused by Rhizoctonia solani. Turns out that the causal agent is different. It's Rhizoctonia cerealis, which previously was thought to just be a different strain of Rhizoctonia solani. This one has binucleate hyphal cells, a different sexual stage, like I told you, when that one has been induced. It's a ceratobasidium. Attacks primarily cool season grasses, but can be a problem on Zoysia, where Zoysia is cultured in cool weather environments like around Chattanooga or northern Arkansas, places like that. And it's a cold, wet weather disease, almost the same conditions that are conducive for pink snow mold are conducive for yellow patch. Symptoms of yellow patch: we never really see turf killed by this fungus, but it can be unsightly, and these patches can really persist because when they're coming on, when these symptoms are coming on, the turf is barely growing or not growing. It's almost in a winter dormancy stage. So if you get symptoms, you're looking at them a long time. You're basically waiting for that turf to grow out of the symptoms, which it's not going to do in real cold environments. Because of that, your best control with yellow patch is always preventive. Curative control, you might kill that fungus there, but you're just getting revenge. You're not healing those symptoms up. So preventive control is really, really necessary. There's another mild infection like we might see in the south. And of course, this occurs on overseedings as well. Poa trivialis gets this readily in the wintertime. Another patch, more of a solid patch, although this one does tend to occur more in a frog eye pattern, as you were seeing in the previous slide. Then here's an old slide of a Zoysia lawn up in northern Arkansas with yellow patch, occurring in October, November time period. Better slide showing the patches, frog eye patches. Looks, a lot like necrotic ring spot. In Kentucky bluegrass, it'll look a lot like necrotic ring spot. And even occur in somewhat the same timing as necrotic ring spot. You'll get leaf lesions in Zoysia, and those leaf lesions really look like dollar spots quite a bit. These little dots that you see here are actually sclerotia, the Rhizoctonia that are being formed. And I showed you, or talked at least about microscopically how we would distinguish this from sclerotinia, from dollar spot. But this one is white in culture, whereas Rhizoctonia solani was brown, has a different sexual stage when it's formed, and then, we even have different AGs that are designated for the binucleates, which some of which are, pretty good pathogens of other plants. There's our DNA stain showing the cell wall here and 2 nuclei clearly. So diagnostically, we get a sample in the lab. It's been very cold weather. It's bent grass or it's Poa trivialis. We get a digital photograph with it showing those yellow rings, and just incubating a little of that overnight, we can get some fungal growth. Put some stain on it. Look at it under a fluorescence microscope. If we see these binucleate hyphal cells, we can be pretty darn sure we're dealing with yellow patch and not brown ring patch, which will look almost exactly the same but it's going to be multinucleate. These fungicides are actually labeled for yellow patch or cool weather brown patch, as you see here. You can recognize some of these fungicides also as being very strong against Rhizoctonia solani. And here we still have our T-methyl formulations for yellow patch. What tends to work the best, I think, are the systemic fungicides, the ones that are xylem mobile, things like Heritage. The Reserve ought to work well from the triticonnazole. The Disarm formulations should work fine. ProStar is excellent for this. Kearney is very good, etc. Now these that are localized penetrants like a foam, probably less effective than the systemic materials. Now other binucleates that occur are out there in patch. There's been some research projects that have looked for these things, and it turns out that they actually predominate when you look for them on patch or in patch. They haven't been characterized genetically very sufficiently, but what has been done has shown that they seem to be very low or avirulent, no pathogenicity. So because of that, there's been at least one research project where they were looked at as biocontrol agents. And they were decent biocontrol agents, but of course fungicide use is going to kill these things as well as your pathogens. Here's what they look like culture, kind of white or tan in culture. So there's an area of research that's been ignored lately. And we have other things to do with the bona fide pathogens, like this next disease, Rhizoctonia leaf and sheath spot. Okay? So the causal agents here, Rhizoctonia zea and oryzae, and you might recognize ZEA is the genus of maize or corn, and ORYZA is the genus of rice. And it turns out that these fungi were described originally back in the 1930s on corn and rice, respectively. These organisms tend to predominate in very hot weather, even warmer than Rhizoctonia solani. So when we've done oscillations in bent grass, we can start to pick these up when we get air temperatures that are consistently in the 90s up to 100. And we also have a problem in Bermuda grass from some strains that show up in late summertime on Bermuda and are causing some real aggravations, particularly in some of the ultra-dwarf Bermuda grasses. We do get leaf lesions with these. We don't think that they are important as root rots. We'll get a blight of the leaf tissue, given this kind of a reddish, yellowish coloration. And some of the symptoms actually can look very similar to symptoms brought on by Pythium root rots and root dysfunction. With the warm season grasses, we don't see so much leaf lesions as we do just blight. And here's an example. It looks like yellow patch. If you look at that photograph there, the difference is yellow patch is in cold, wet weather, and this photograph is taken in the middle of the summertime on bent grass. Really, really hot, and in fact, too hot for brown patch. Rhizoctonia solani will actually subside with some of these heat-tolerant bent grasses when it gets really, really hot. I think you might see a hint of a smoke ring here on the edge of that patch. Here's another one in bent grass, kind of a bronze solid patch with a active margin here. Very hard to tell that from some of the Pythium, unless you get it under the microscope, and obviously you'll see the mycelium of the Rhizoctonia if you look for it. And then here's an example of it attacking some young seedling bent grass being established in warm weather, causing symptoms that would just be typical of brown patch. We also pick it up a lot in overseedings, and this is Poatrivialis over Bermuda grass. We'll start to pick this disease up about now, right on through transition, typically. In the past, this has been referred to as leaf and sheath spot caused by Rhizoctonia zea. There's a couple of reports that some of this is actually brown ring patch, which makes sense because we're talking about poatridialis here. But that's an area where we need more identifications. And it may be mixed infections. When you get really humid conditions, this will look a lot like Pythium blight, which is, you know, if you were to walk out there and see this after a real wet night of rainfall, see that early in the morning, you'd probably be grabbing your Pythium fungicides. But you put that under the microscope, and it's not Pythium, it's Rhizoctonia zea and or cercinata. Diagnostically, that's kind of part of what we're talking about here today. Those that have experience looking at these fungi as they're growing out of plant tissue can really recognize some patterns. This is actually Rhizoctonia cerealis here that causes yellow patch. This is Rhizoctonia zea, and Rhizoctonia cercinata would look the same. You can see the difference here with all these vacuoles that are here inside the mycelium, and that's typical of that young, actively growing stuff. You see the cell wall there right after the point. There's the constriction. Got the 90-degree angle. You know, everything looks the same. Here's the constriction in the cerealis, pretty much a 90-degree angle, and the septation, that's the typical pattern. If you were confused and you wanted to throw a little DNA stain on this and look at it under a fluorescence microscope, this one would be multinucleate, this one would be binucleate. So labs that have that capability should be able to tell you within about 48 hours, and that's without using modern DNA techniques. Now, zea looks like this in culture, totally different, kind of salmon pink in culture. These little tiny sclerotia are formed, which have been sieved out of thatch. We find them in nature. And here's some different patterns of these sclerotia. They're embedded in cultures. You can notice that these are a little larger than the ones over here. And in general, these larger sclerotia are characteristics of the Rhizoctonia cercinata, where these little tiny ones are more likely Rhizoctonia zea. But to distinguish those really does take DNA techniques nowadays. And here's an example of a ladder, that one of the guys that works with me, Dr. Hannafy Foley, constructed based on some of our isolations from different grasses. These are based on the DNA sequence of certain genes, and all that's telling you is that all these different strains of Rhizoctonia zea are grouped together genetically, as they should be. Rhizoctonia rhizae very closely related to it. Even cercinata related, but somewhat separated, which is true. We're even seeing the diseases be separated based on temperature optima and possibly even some host range. Compared to Rhizoctonia solani, different strains from bentgrass, and Rhizoctonia cerealis down here, this should be down a little lower here, showing you that these are bona fide different biological species, and of course, different diseases. Now, I've kind of referred or alluded to this through this webinar that zea and rhizae and cercinata are not affected by thiophenic methyl fungicides. Even though we see T-methyl on some of the labels, nowadays some of these labels from Bayer, BASF, Syngenta especially, are distinguishing these diseases and these fungi, which is good. And Cleary's is doing it as well. So you're not going to see T-methyl labeled for these diseases now that we're distinguishing the diseases. And basically, let me explain this slide here. This is a tall fescue plant that I had inoculated a few years ago with Rhizoctonia zea, and this plant had been treated with a high rate of Benamil, which is a benzimidazole fungicide, and thiophenic methyl is closely related in the same chemical class. And we've even seen this experimentally in some of our field research where we've increased Rhizoctonia leaf and sheath spot during heat stress intentionally by putting thiophantic methyl in the middle of a program. So here's some old work that was done about 12 years ago, and you can see the fungicides banner went on the first part of June, followed by Daconil in mid-June, followed by a high rate of 4 first part of July. In the second week in July, Cleary's 33-36. Not even a real high rate there. Followed by Heritage, followed by ProStar. Good fungicides. All of them good fungicides for Rhizoctonia solani. And here's the program. Everything is doing great with this program 7 that I just went over. We're getting great disease control until that T-methyl application went on, and then suddenly the apparent brown patch accelerated. We did lots of isolations from this disease, and it all came out as Rhizoctonia zea. Whereas a different program, I didn't show you this, but this one had thiophantic methyl as one of the first sprays. And in these first few weeks through here in the summertime, Rhizoctonia solani predominates. Whereas in the hot heat of the summer, Rhizoctonia zea predominates. This kind of phenomenon, I think, is not uncommon in real heat stress environments where we're growing cool season grasses. Distinguishing leaf and sheath spot or Rhizoctonia zea on labels is now being done, and you can see some of these different fungicides that are labeled specifically for leaf and sheath spot or Rhizoctonia zea now, which is good because they're bona fide different diseases. It's good to recognize that. And you do not see T-methyl there on that list. Now finally, brown ring patch. This is the latest disease that we're distinguishing on cool season grasses caused by a different Rhizoctonia. It has been called Waitea patch, but for reasons I've already discussed, we would prefer to use the term brown ring patch. Then the taxonomist can have at it. We'll still be talking about brown ring patch. Here's a famous photograph that Frank Long took. Looks just like yellow patch, doesn't it? This is on Poa annua down in the San Diego area. The difference is photograph was taken in May, so some fairly mild weather conditions, but certainly not cold and wet when you would tend to have yellow patch. So we were seeing these things showing up around and about, and wondering if some of the yellow patch was just persisting, and possibly with the trend not to fertilize much, some of these symptoms were persisting. But originally, some research in Japan distinguished brown ring patch, identified Rhizoctonia cercinata variety cercinata as the causal agent, and then later Frank, the following year, Frank distinguished this in California. These are some photographs on bent Poa greens from Ohio. Mike Bain, Carl Dannenberger took these photographs, where the disease is preferentially attacking the Poa and not the bent. And another set of photos from Bain and Dannenberger from Ohio. And then some photographs from Massachusetts, from John Kaminsky and Connecticut, Rhode Island area, all with the same symptomology, all showing up in the late spring. And it has also been found in the early fall as well. So mild conditions. This is a map where it's been officially reported in some way in Plant Disease Reporters, Plant Disease Journal, somewhere, or Plant Health Manager. Doesn't mean it doesn't occur in these states. For sure it does. It's been identified up in Canada and various places. And it's been positively identified, at least the causal agent, Rhizoctonia cercinata, on overseedings in South Carolina, Florida. This was an official report. and officially reported on Poatridialis and on ryegrass in Southern California by Frank Wong. So we will see brown ring patch being identified, I believe, on some different grasses. The more we look, we're going to see it. It has the same sexual stage as Rhizoctonia zea and oryzae. Fungus is Waitia cercinata. Multinucleate instead of binucleate hyphal cells. Sclerotia slightly larger and tend to be slightly larger than Rhizoctonia zea. And it, in common with zea and oryzae, it's insensitive to benzimidazole fungicides. Now this is debatable, I think. Maybe more damaging to POA or have a host preferences to POA. That's the pattern we've been seeing lately. But this past year it was reported on bentgrass, and I have some isolates from bentgrass that did not have POA in the greens. Where it was reported on bent was in New Jersey out of Bruce Clark's lab. Of course, this is a moderate weather, humid weather disease, and it has been controlled with fungicides except for T-methyl. So those are good things to know. Here's one fungicide trial that Frank Wong and Larry Stow and Wendy Glarner did in 2006. It was a curative trial. And the main points here are that the T-methyl here is no different from the check, basically. So that was some field evidence that this T-methyl is not going to work. And in this case, Compass didn't do quite as good a job as some of these other fungicides. And these are not statistically significantly different here. But you recognize the names ProStar, Heritage, Endorse, Medallion. Even some of the DMIs like Banner and Tritaconazole and Metconazole are doing a pretty good job with brown ring patch. In addition to light fertilization, that's reminding myself to talk about that with brown ring patch because that's part of it is trying to get the host to grow out of these symptoms a little bit. These are fungicides that recently have been labeled for brown ring patch. So companies are recognizing this as a bona fide different disease with the fungicides that you see here. And with that, I'm back to what I told you I was going to talk about. So we went through brown patch, we went through leaf and sheath spot, we went through brown ring patch, and we went through yellow patch, which gives you, I think, a pretty good overview of the rhizoctonias that are occurring on cool season grasses. You add large patch and leaf and sheath spot on Bermuda grass, and you have the whole picture as far as we know about it today. And with that, I think I'm about 1 minute over time, which pretty good for me, being that close. So I'll turn it over to the moderators, and I'll try to answer questions.
Very good. Dr. Martin, first question, will yellow patch weaken turf and cause problems during the summer if it's not treated?
I've seen some severe yellow patch where it's occurred under snow covers where you get a little bit of a sinking, a little bit of thatch degradation with it. But by and large, it's really a minor disease from the standpoint of damaging the host. I don't think it's going to persist and cause problems. We generally see it grow out of those symptoms. Fungicides would be necessary if you had chronic problems with it and you didn't want to be looking at those symptoms all winter long, primarily.
Dr. Martin, with some other diseases like anthracnose, dollar spot, gray leaf spot, we have resistance issues to repeated use of fungicides. But that doesn't seem to be the case with Rhizoctonia. Can you elaborate on that and what makes it different from fungicide resistance?
What makes it different? We're not really sure. It's certainly all of these Rhizoctonias have a saprophytic phase, and I think some of them, most of the diseases where we have more of a resistance problem, are really more bona fide obligate type pathogens. There's a little more intimate association genetically with the plant. But to be honest, I don't think anybody really knows. Just historically, we've had no problem with resistance with these, and it's a good thing with the reliance on strobilurins and DMIs that we're seeing now, because we're certainly seeing it increase in diseases like anthracnose and dollar spot. So, Lawrence, I really don't have a great answer for that, other than historically, even with some of the specific fungicides, we haven't had a problem. Now, the T-methyl thing is not a resistance issue. It's just a lack of sensitivity. There's just something fundamentally different about those fungi that are not affected by T-methyl.
And I've got another question. This is regarding large patch. And the question is, when's the best time, soil temperatures, type of fungicides, and number of apps to really effectively control large patch and Myrzorrhea?
Yeah, Myrzorrhea or any Zorzia, as far as we know.
Yeah, I know you didn't really address that today, but we do have that question. So if you take a few minutes to talk about that, that'd be great.
Yeah, large patch is caused by a strain of Rhizoctonia solana, and it's attacking the warm season grasses when they're weakened. Hopefully you kind of got that idea, through this talk on the cool season grasses. But we typically had problems with large patch in more transition zone environments, or where those grasses really slow in their growth going into winter. And there's some differences in susceptibility. Zorzias seem to be quite susceptible, but we're pretty sure the initial infections are taking place from inoculum that's right on the stolons and the rhizomes, and it's affecting the shoots, the lower basal leaf sheaths. In my environment around South Carolina, it's going to be in early September, probably, even late August, some infections. As you start to get colder, the symptoms may or may not be produced. On Zorzia, a lot of the time the symptoms are not even seen until the spring of the year, but we do know that the initial infections are occurring in the fall. So those are actually the key places to apply the fungicides. And I think they're looking at data, my own experience with it, it kind of makes sense that the penetrant fungicides, the strobilurins, Prostar, some of the DMIs, do the best job, and I think the key applications are the initial ones. So in my area, that would be around the first part of September. 28 days later or so, another application is going to tend to give you better control than one application in the fall. And then the Cadillac treatment would generally be 2 in the fall and 1 in the spring to try to clean up any disease that went through those infections. Now, 2 years ago in the Atlanta area, we saw large patch go through everything. The fungicides that were down, so you just got to be aware sometimes Mother Nature will overwhelm the ability of the fungicides to do it. And that occurs with all diseases. We shouldn't really be too surprised by that. But that's the timing, I think. And I would rank them 1, 2, 3 in their importance. The first one's September, the second one in October, 30 days later or so, and then the third one in importance would be the spring. If you didn't do those fall ones, you're going to see a lot of disease in the spring. And basically, again, you're just kind of getting revenge on those, stopping those patches from expanding, but you're not going to get lots of healing until temperatures warm up.
Next question came in was, are the chemicals in the group, like the Heritage, going to be more expensive than the chemicals in a group like Chipco 26GT?
You probably answered that better than me, but the newer, more specific fungicides tend to be more expensive. I don't know if it has to do, this probably has to do with marketing as much as anything. Sometimes it has to do with cost of production of these fungicides, but, I would think that chemical companies are going to base their prices on demand and on value added. So I told you that some of the dicarboxamides are giving you about 2 weeks of control. They're good brown patch materials. They're not great brown patch materials. The great ones tend to cost more because they give you great control instead of good control. That's value added.
All right. Well, it looks like that's all the questions. So, again, I want to thank everyone for joining us. Just a reminder, you will, if you're applying for the GCSAA CEUs, you should get that information within the next 6 to 10 days. And our next sessions are posted, and we look forward to you joining us in the future. And I want to thank Dr. Martin again for his time today and enlightening us on Rhizoctonia and brown patch.
Welcome. Thank you.
Ladies and gentlemen, that does conclude the conference call for today. We thank you for your participation. You may now disconnect. Have a wonderful day.